Hi, I'm Sarah and I'm a PhD student at Imperial College London. I'm developing a technique to help assess the sunlight degradation or photodegradation pathways of agrochemicals, that includes pesticides, on a model plant leaf surface. The intention of this study is for these techniques to later be used as a tool to help scientists to better understand how these compounds respond to sunlight in their design and development of efficient new agrochemicals. Now, the key thing to note in this work is that sunlight is extremely energetic. It provides enough energy for the whole Earth and almost everything on it. And that's without even considering that only 1 to 2% of the available energy is used. But with great power comes great responsibility. And the sun's energy can also be destructive. Chemical bonds are made from a pair of electrons, and when a chemical compound is in sunlight, these chemical bonds can absorb the light energy and be split in half, leaving two fragments, each with an unpaired electron. We call these fragments radicals, and radicals are very unstable. Once formed, they need another electron pronto. As such, these radical fragments quickly go on to react with the surrounding environment, and on their hunt for another electron, will rip apart other chemical bonds and take an electron, thus forming another radical. This causes a chain reaction of radical formation until finally all radicals have a pair. Now, this makes analysis of photodegradation reactions particularly difficult. The speedy reaction and often collapse of these radical fragments means that many of the most common analytical techniques cannot detect these species. Instead of measuring the radicals, the techniques can often only detect the final product of the chain reaction, which may be far removed from the initial radical fragments that were formed after the chemical bond was split by sunlight. This is where electron paramagnetic resonance, or EPR, comes in handy. A paramagnetic compound is another word for a compound with an unpaired electron, like the radical fragments we've been talking about so far. So EPR is designed to observe radicals. That must make it really easy to observe these reactions with EPR, right? Not quite. These radical fragments are still exceptionally short-lived, which, even with EPR, makes it incredibly difficult, but not impossible, to detect them. In my project, I'm first trying to directly observe these radicals so that I can see which bonds in an agrochemical are broken when sunlight is absorbed. Then, I want to see how this is affected when you introduce some of the complexities of the natural environment. After all, this is about agrochemicals. I need to know how they'll behave on a leaf surface. With the understanding that this research could provide of the photodegradation pathway of agrochemicals, it could be possible to control these reactions to make agrochemical products that are more efficient. This could help crop production using agrochemicals to use fewer resources whilst continuing to provide enough food for our growing population.